Good afternoon, everyone. Thank you so much for joining us today. My name is Emily Silva, and I'm the NECAN coordinator. I'd like to welcome you to the first webinar in the NECAN Sea Grant webinar series. In 2016, the Northeast Sea Grant Consortium, in partnership with NOAA Ocean Acidification Program, funded 4 research projects to address the impacts of ocean acidification on key resource species in the Northeast as an aid to assist coastal communities in adapting to current and future OA conditions in the region. The research and observational priorities defined in the publication of a NECAN paper in Oceanography magazine, Ocean and Coastal Acidification of New England and Nova Scotia, were used in the 2016 request for proposals. 4 projects were chosen and funded in Connecticut, Maine, and New York, and each of these webinars will focus on one of those projects, their successes, challenges, and results. The full webinar schedule can be found on the NECAN website, and each of these will be recorded and posted there as well. Throughout the presentation, if you have any questions, you can submit them through the Questions tab on your GoToWebinar control panel, and we'll go through each of those during the Q&A session following the presentation. Today, we'll be hearing from Dr. Hannes Baumann on the unusual sensitivity of northern sandlands to acidification and warming. Dr. Baumann is an assistant professor and leader of the Evolutionary Fish Ecology Lab in the Department of Marine Sciences at the University of Connecticut. For the past 10 years, his research has been primarily focused on natural and man-made reasons for ubiquitous changes in marine fish abundance, growth, and population characteristics. And he is particularly interested in studying principles of short and long-term adaptation of marine organisms to ocean warming, acidification, and commercial fishing. With that, I'll hand things over to Hannes, and he can take you through his presentation.
Thank you so much, Emily, for that kind introduction. And thanks to all of you for taking some busy time out of your schedule and tuning in to this webinar. This is a first for me. I've never presented science in that kind of form, so I'd be very curious to hear from many of you, either via email or through the website or via Twitter, if you like how this whole thing went down. So I also want to acknowledge my co-authors and collaborators on this project, Dr. David Wiley from the Stellwagen Bank National Marine Sanctuary, and Chris Murray, who until very recently had been my PhD student here at the University of Connecticut, and who is now doing his postdoctoral research as of a few weeks ago at the University of Washington. So the study and the story that I'm going to tell you more or less started in 2015, and by now it's 2019, and within a few weeks this part that I'm going to talk about today will hopefully appear in the Journal of Conservation Physiology. So please stay tuned for that. But here's a visual, basically, of what we are going to talk about in that publication. And both Dave Wiley and Chris Murray are authors and myself on this study. So the story I'm trying to tell begins more or less in 2015, when we not only realized that the folks up in Scituate at the Stellwagen Bank National Marine Sanctuary were wonderful colleagues and good collaborators, but also that they shared our passion for this species, the northern sandlands, and the opportunities and that arose basically from, or that you recognized from investigating this species. As Emily already said, we were fortunate with all our enthusiasm to convince the reviewers and the panel of the Northeast Regional Sea Grant Consortium to be one of the 4 funded projects in that funding cycle, and basically started us off for good here in 2016, all the way through the beginning of 2018. And then some additional funding came about that is still ongoing from the Bureau of Ocean Energy Management, which I would like to acknowledge here too. You can see in the background Chris Murray on our NOAA boat, on the NOAA boat Aug, sifting through a bag of sand, which is our main way of catching sandlands during this project. Now, the whole story, as some of you may know, plays out in that enigmatic area just north of Cape Cod called Stellwagen Bank National Marine Sanctuary. It's an area that is in the southern Gulf of Maine, and as many of you are undoubtedly aware, this is an area that has over the past decades undergone dramatic changes for the ocean, particularly in warming, and masked by some changes also in salinity, acidification. Now, this is an area that attracts hundreds of thousands of visitors every year, and these people are coming because they want to see iconic wildlife, iconic marine piscivores such as whales and tuna and the charismatic seabird fauna that is found there. So when we talk about why study sandlands, the answer, there's two parts to that answer, and the first and primary one I'm giving away, of course, here, and that is because sandlands literally feeds almost anybody with a bigger mouth on Stellwagen Bank. Most notably and most iconically, a large population of humpback whales who are actually finding out where these sandlands are and are co-occurring with them on a yearly basis. So where the sandlands are, the humpback whales are. But humpback whales are not the only ones. A charismatic and lots of species of seabirds feed on sandlands, which are very nutritious compared to other forage fish, but also cod, tuna, sharks, like dogfish and other animals. So everybody is eating these fish, and therefore they are rightly called the backbone of the National Marine Sanctuary. Without sandlands, undoubtedly, there would not be this charismatic fauna. So when we talk about a resource that is truly important, not just for the animals, but also for humans that enjoy them, sandlands is a very important and foundational species. So the idea to investigate how these fish will fare under a future conditions of a warmer and more CO2-rich ocean has a primary importance. The other part of that answer I will leave for now and will come back at the end of this talk. Now, I've been studying the effects of ocean acidification, particularly on fish, for almost a decade by now. And one thing is very clear when it comes to these organisms or fish in general, is that their highest vulnerabilities to elevated CO2 levels and to temperature lie in their early development, at their early life history stages, meaning the developing eggs, the embryos, and then the early larval stages. We know from the literature for quite some time that once fish are actively swimming during their juvenile and their adult stages, they are relatively able and very well equipped to deal with high CO2 levels. And that's why our focus and the focus of many research groups throughout the world is to understand what does high CO2 do to these early life stages. Now, there are a lot of modeled species where you can go to a website or to a fish store and can try to buy their offspring from some commercial supplier. But when we talk about non-modeled species like the northern sandlands, there is simply no alternative to go out physically and catch them and try to obtain their eggs and their spawning products by yourself. And that is why our collaboration with the folks up in Scituate from NOAA, from the Stellwagen Bank National Marine Sanctuary, was such a great fortune, because without their help, we would not be able to do that. What you see here are fertilized eggs of northern sandlands, which we obtained in a classical way, as you can see here, with these spawning dishes where the eggs and then the sperm of the females are mixed together, and then the eggs and our treatments are starting after that. We did this both at sea, directly after catching them, and because we wanted to hedge our beds, we also did that in the laboratory, where we had a little more bells and whistles and tried to do a few things. Remember, this is not a modeled species, and almost anything that we had to find out about their rearing and all the mundane stuff that goes into doing an experimental research on a non-model species, we had to find out by ourselves. Here you can see us on the NOAA ship Auk, which has been our basis for all the duration of the project and continuing. And we caught sandlands not during the beautiful time in June when the whale action and the tourist action is highest, but when in November, when these fish are actually spawning. This is one of the important characteristics of this fish species. It is that they are winter spawners. And we first had to find out exactly and if sandlands do indeed spawn on Stellwagen Bank. As you can imagine from the title of that project, we found spawning sandlands. Another thing that you can see from this little short video clip here is not only that we managed to get these fish transported live to our rearing facilities, but also that these fish are peculiar in their sort of like their way of living. They are they're alternating between these large schools feeding on zooplankton in the water column, but then every day or periodically, we don't quite understand yet how they decide that they are actually buried in the sediment. And that sediment has to be a fine sand that you can see here right in front of you, of a specific grain size and very little organic content for these fish to actually thrive there. Sand that is exactly like the one on Stellwagen Bank. Now, what we wanted to do, we wanted to employ a system, a factorial experimental system that we have built in 2015, and we used for various research on this and other forage fish species. And what you're looking at the back of this picture here in the Rankin Lab at the University of Connecticut in Avery Point, you are looking at our factorial rearing system, that tarped structure in the back of the blue tanks. Unfortunately, it is very hard to do or get a good picture of that whole system. But the point is that these are 9 independent recirculating units that we can automatically, meaning with a computer program called LabVIEW, control for their CO2 content and their oxygen content and their temperature, and therefore can basically provide and conduct these replicated and factorial experiments. For this project, we chose to work with 3 CO2 levels that were crossed by 3 temperature levels. The 3 CO2 levels, for those who are familiar with the ocean acidification literature a little bit, are relatively common. We are contrasting here an ambient level of about 400 microatmosphere in our with our alkalinity here that equals about 8.2, a pH of 8.2, whereas elevated and high CO2 levels corresponded roughly to the levels that we expect at the end of the century, and a maximum level that is predicted to occur within the next 300 years if humans are foolish enough to indeed burn every single piece of carbon that they can dig out of the earth. we crossed these with these 3 different temperatures based on some long-term data that exist for Stellwagen Bank. You can see this here. These MWRA hydro data are unfortunately collected between February and November, which exactly excludes the period of interest for us here. But with a little bit of interpolation, you can see it's not hard to deduce that temperatures during the spawning time of sandlands are decreasing slowly to about 5 degrees Celsius, which is our low temperature and the temperature that we consider to be the most representative of today's temperatures for sandlands, a higher one of 7 and then a contrast of 10 degrees that basically assumes that it doesn't get cold during their time at all. So these 3 temperature levels were basically chosen because we had some kind of understanding what the temperatures on Stellwagen Bank look during the winter and the developmental period of sandlands. I should note before I show you more that before we started, we had only a very rough idea about when sandlands, northern sandlands in particular, spawn during the time of the year. If you consult the literature on the subject, you can still find that northern sandlands have an extracted or protracted spawning season from the end of October to the end of February. Now, in our first year in 2016, we actually tried to cover this entire window in order to find out when they would actually spawn. In contrast, what we found out, at least for Stellwagen Bank, sandlands do not spawn over a protracted spawning period at all. At the contrary, these fish in all 3 years, and we are talking now 2016, 17, and 18, these fish spawned during a very narrow, about 2 weeks window at the end of November. Because these fish have the unfortunate characteristic of always using their peak spawning at the time of Thanksgiving, we christened them Thanksgiving fish. Now, what you see here is first a graph of the gonadosomatic index of females that we collected in 2017. These are data that are not in the manuscript, they're not published yet, but we basically found that the gonadosomatic index, which is a proxy for the ripening and the time of spawning in fish, increases sharply from the beginning of November to about Thanksgiving, and then drops off precipitously at the end. The same is true for the diameter of the oocytes. What you can see in the back is a micrograph of the ovary of a female, a representative female here, and you can see these large, uniform in size secondary oocytes that are in that ovary. And the only other structures that you can see in here are these little orangey dots above those, which are actually reservoir oocytes that will not ripen any time until the next year. So those are next year's reservoir oocytes, whereas those big dots with those oil droplets and these vitellogen droplets inside, those are the secondary oocytes. And because they are all the same size, that means they are all going to be spawned at the same time. So what we found out was, first, yes, sandlands do spawn on Stellwagen Bank, and secondly, they do not spawn over a protracted window, but they spawn over a very narrow window at the end of November, and afterwards they don't. Now I show you some results from our pilot trial. Again, I stress that these fish are non-model species. We are basically the first ones who investigate the sensitivity to CO2 and warming in not just northern sandland, but sandlands, period. There are about 8 sandland species. They are all distributed in the northern hemisphere of our Earth, and all 8 of them are as important as the northern sandlands in terms of their role in the ecosystem, being basically food for large, higher trophic levels. So our pilot trial in 2016 reared these fish from our fertilization at sea. And what I show you here is the survival of the embryos. That means how many have hatched in the different CO2 treatments, contrasted with the, in this case, two temperature treatments. We have included that 7-degree treatment only in the next year's trial. What you can see is that at 5 degrees Celsius, the overall hatching success was about a third, a little better than a third. And at the ambient conditions, whereas at the end of century predicted CO2 levels, the hatching success was less than half of that. And then at the maximum CO2 levels of about 2,300 microatmospheres, we can see that only a 1% hatching success was registered in this pilot trial. Now, these were unreplicated trials. They were again used to get us familiar with the methods and with all our rearing protocols. But what you can already see in this particular trial is that if you increase that temperature by 5 degrees, which you might argue is a rather large step, but remember, these fish more or less spawn at that time, at that temperature, and then the temperature decreases throughout their developmental season. But if you keep these fish developing under constant 10 degrees Celsius, and you register about 2% hatching success in that particular year, and again half of that in with the elevated CO2 conditions, and there was not a single hatchling at 10 degrees Celsius and 2,300 microatmospheres. So what we learned about these initial pilot trial in 2016, we were surprised by the severity of this apparent CO2 effect. Apparently, the hatching success of these fish is very, very sensitive to increased CO2. But as many of you at that particular moment will probably think, and if there would be a live audience, maybe some of you would raise their hands and would say like, well, that is a pilot trial. If you really think you have some story to tell here, you need to replicate it, show us. And that is what I would say myself. And I would say like, well, that sounds like there is something here. Now do it again and see if you can replicate these results. So what we did in 2017, we tried to do everything right. And as many of you who have worked experimentally know, you never do everything right. But I believe we did most of things right. So we hit these fish at the height of their spawning season, which is good. We tried to both fertilize them at sea, so under the rocky condition of the Auk, and in the lab. 2 days after we caught them, we strip spawned them, those that we have transported in the lab. And we conducted these things at the temperature and the 3 CO2 levels that were similar to the pilot trial. What you're looking at here now at these upper panels are cumulative hatching distributions. So it says hatching success. You could say that this is the cumulative percentage of fish that hatch. If you focus on the blue line first, you can see that starting at about 5 weeks post-fertilization, we see this large spike in hatchlings emerging. And then we have a number of sort of like subsequent hatching peaks here that over the course of another 3 weeks are basically hatching afterwards. After 60 days post-fertilization, there's basically nothing that is hatching. Everything that would have hatched has hatched at that point. And you can see that under these ambient CO2 conditions and 5 degrees Celsius, we see about 60% of the fish hatching. Now contrast this with the CO2 levels, the elevated CO2 levels of 1,000 and 2,100 microatmosphere, and you see that this dropped by more than a third. About a third of the fish that have hatched at ambient hatched under high CO2 at 5 degrees Celsius. If you go to the 10 degrees Celsius, you see that even under ambient condition, the hatching success decreases precipitously by a little more than half, whereas under high CO2 conditions, you see a further decline to less than 10% and about 2% hatching success. So the overall picture here is that the high sensitivity of hatching success to the high CO2 sensitivity of these fish is replicated from 2016 to 2017. I'm showing you lines here that are means. These are means of 3 replicates. For clarity, I'm only showing you here the mean, basically, responses. These lower panels here are an interesting sort of addition, because the way we quantified that response is by literally checking every day for hatched fish, for new hatchlings, and then we scoop them carefully out and we put them into a separate container, and we found that way we found that while the overwhelming majority of fish under ambient conditions hatch within a very narrow peak. You can see this very clearly both at 5 degrees C, but even better at 10 degrees C. So they all hatch basically at the same time. The hatching of the high CO2 fish seems to be kind of delayed and trickling out more over time. In fact, when we look at this more and more, we're becoming intrigued in this pattern most, because it seems that the fish somehow have trouble hatching under these high CO2 conditions, and because they can't get out of their eggs, they kind of trickle more and more out of it. So there's a delay in hatching, and there's a large effect of a large mortality effect or survival effect of both CO2 and temperature. These were the fish that we fertilized at sea. Remember, that's a fish that has this very narrow spawning peak, and I mentioned that because, as it turns out, even though we had better methodology and there was not a rocky boat in our lab, but just the fact that we kept these fish in the lab for 2 days apparently had a dramatic effect on our ability to fertilize these eggs. What you see here is exactly the same kind of graph, but you can see that that blue line only rises to about 16% hatching success at the end. The differences between the CO2 seem to be similar, and the hatching patterns seem to be similar, but we believe that just the fact that we kept these fish for 2 days after their capture and then tried to fertilize them, strip spawn them, the way we do this regularly with other fish species, this had a dramatic effect on the fertilization success of these eggs. So note to ourselves, if you want to do this right, you have to fertilize them when you get them out of the water. Now, we were still not satisfied with all of this, because I'm a strong believer in repeating experiments to really make sure that we are not interpreting some artifact. You never know if an experiment has inadvertently introduced a cofactor that you don't think of. So the best way of making a conclusion robust is to repeat these experiments again and again. And here we have the 3rd iteration, and given what we know about how much it takes to actually get these fish and how much it takes in terms of conducting these experiments, I'm very, very grateful to my former postdoc, Dr. Emma Cross. She is now a newly minted faculty at the Southern Connecticut State University. My current master student, Lucas Jones, and Chris Murray, all these three basically pulled it off and did a third year trial in the end of 2018, just to make sure that what we see here is actually a robust pattern. If true, this would indeed mean a mandate to understand much more about this fish. As you can see, at 5 degrees C, we see at 400 micron the best hatching success that we've seen in all the 3 years, about 70% here of hatching success. It seems that we have gotten a little better over time in how to do these experiments, and then almost as if drawn by desire, you can see these fish at 1,000 microatmospheres going to or below 50%, and then at 2,000 microatmospheres we have not even almost 20% of hatching success. So again, a large effect, and I stress that this is for a fish, this is a very unusually high effect or large effect. At the 10 degrees C treatment, you can see that we tried a few different things with our system here. We of course included the 400,000 and 2,000 microatmospheres, but we were also interested in where exactly sort of is that biggest sensitivity, and we included a few treatments between 400 and 800 microatmospheres. We included a 600 and a 700 microatmosphere treatment, and you can see that those are all pretty much at the same level here. Interestingly, at this level, we do not see a large temperature effect, but we again see a predictable effect of CO2. In conclusion, what we find about the embryo sensitivity to high CO2 in this fish species is a robust pattern. This is, I believe, one of the most CO2 sensitive fish species tested to date. Here you can see me and Chris in our lab in front of our image analysis station, and we were very interested in not just counting the embryos that hatched, but also look at the embryos under the microscope. And you can see here a figure that will appear in the paper as you can see it. What Chris did in this particular aspect, he measured 4 different things in these newly hatched larvae. One of them, there's the somatic body area, and there's the hatch length, and there we measured the oil globule area and the yolk sac area, and then we used the principal component analysis to basically reduce these 4 variables to 2 principal components. You will not be surprised to find out that the somatic body area and the hatch length grouped together, they formed the first principal component, and those oil globule area and yolk sac area, which are really proxies for how much energy that larvae brings into its critical transition from endogenous to exogenous feeding, that was the second principal component. You can see here that the ambient conditions at all temperatures had the largest body size, and they had the biggest endogenous energy reserves, basically. And you see an effect of temperature, positive effect of temperature on these body size metrics, and a slightly negative effect of these endogenous energy traits with temperature, which makes sense. But again, here you can see there is an interaction between CO2 and temperature, because at the highest CO2 levels, that increase in body size in that first principal component precipitously sort of levels off at the higher temperature and does not increase like in the other 2 temperatures. So as a take home for today, I'd offer these 3 points that you have heard, and hopefully I'm able to convince you, was able to convince you that these are very large effects for fish, particularly when it comes to these fitness-relevant traits of growth and survival. If, to our knowledge, and looking at the recent meta-analysis by Catano, we believe that sandlands, northern sandlands, might be the most CO2 sensitive fish species that has been tested to date. We see synergistic negative temperature effects, but not in every year. And sometimes there are additive, sometimes there are synergistic negative, but there is no question that a high temperature and high CO2 level by far elicited or results in the lowest survival of these embryos to hatch. And that, of course, given sandland's role in an ecosystem, should trigger at least some concern and some precautionary approach here, that anybody who believes that we can maybe exploit this species, or that we should not worry about that, might be mistaken. And it might be that this particular backbone of the Stellwagen Bank National Marine Sanctuary is more sensitive to a future high CO2 ocean that we have realized, and therefore all the bigger piscivores that we love and that we love to take photos of might be affected too. Of course, if you are finding a result like this, the more interesting things are the mandate that comes with it to understand why do we see these effects. I already mentioned that we believe there is a mechanism here that prevents these fish to hatch, and that is something that Chris Murray will at the West Coast try to get a better handle on here. I'm personally most interested in the question, well, this species is distributed between New Jersey and Greenland. So is there any geographical differences in these in northern sandland sensitivity? And of course, are other sandland species equally sensitive? And what about acclimation and adaptation? That is the $100 million question of ocean acidification science. We know that CO2 is changing gradually over dozens of generations of sandlands. Are these fish perhaps able to plastically acclimate to it, or even genetically adapt? These things need to be investigated to further conclude what risk or what vulnerability that species has to CO2. But the first step is made, that is basically shown that, hey, if these conditions of in 100 years would actually happen today, these fish would be in deep trouble. All right, and now just for the rest of the last few minutes here, I want to quickly come back to that initial question, why study sandlands, and introduce you basically why we got scientifically so excited and believe that sandlands is a perfect contrast to the fish species that we have mostly studied to date. I mentioned it's an offshore spawner. It's offshore, meaning more not at the coast. It's a winter spawner. It develops very slowly, and that contrasts almost perfectly to another forage fish that we have worked with for almost the last decade, which is the Atlantic silverside, a species that inhabits nearshore habitats, very shallow nearshore habitats, that develops fast and gets it spawned in spring and summer under warmer temperatures. And those of you who are familiar with this field will not be surprised to see, of course, that the Atlantic silverside in this biologically productive nearshore areas is experiencing a much larger short-term variability in CO2, or here by proxy pH conditions that we measured in a nearby cove here, Mumford Cove in Connecticut. Conversely, the offshore area of Ammodytes dubius, the northern sandlands, particularly in winter, is characterized by a almost constant CO2 and by proxy pH levels in the ocean. And if we assume that high CO2 tolerance in organisms comes at a cost, we basically predict that one fish species should be very sensitive and the other one should be rather tolerant. So what you see here is the last figure from the paper, and it basically contrasts exactly that from our almost a decade of work on CO2 and the Atlantic silver sites. We look here at a metric that is called the log transformed response ratio, basically a metric of the effect size of CO2, and on that trait embryo survival, so survival to hatch. And we can see that overall, over the entire experiments that we have done on that silver side, we find that yes, CO2 has a effect, a large effect, not a large effect, a negative effect on the embryos of that species, but that effect is rather small, about 10% reduction in embryo survival due to exposure of about 2000 microatmosphere. Contrast that with about an order of magnitude higher effect size in northern sandlands. And this prompted us earlier this year to formally try to wrap our head around the data and suggest that not just in fish, but in many organisms, we might be able to think better about CO2 sensitivity in the ocean by taking into account the short-term variability that is currently present in these habitats. And there's many gradients in nature where CO2 variability changes predictably, but one of these gradients is the gradient from productive nearshore areas that see CO2 fluctuations between up to 5,000 microatmospheres on a diel basis in the most productive summer month, all the way to the open ocean where CO2 is nearly stable. That means first, of course, that the anthropogenic acidification signal strengthens from nearly undetectable, almost undetectable in these nearshore areas to clearly discernible and significant in the average open ocean. And we believe that if you look at marine species in this way, you can see that the species who are developing in offshore stable CO2 areas, and particularly those that have a slow development, are those that are most sensitive to CO2. The trouble is that most of our empirical data comes from those nearshore species, and we therefore suggest that if we really want to understand who is most vulnerable or who is first vulnerable to ocean acidification, we need to retrain our focus on species that are mostly adapted to these stable oceanic conditions. With that, I want to thank you all for your attention, and I want to plug this and basically make the case that the scientific interest in sandlands, particularly in these sand shoals along the U.S. And Canadian Atlantic coast, goes way beyond their CO2 sensitivity. Two other projects have basically been funded over recent years, one of which I already introduced, that try to understand more broadly questions about connectivity of sandlands and their relationship to the resource of these sandbanks. And I'll leave you with a little stretch I crudely threw together a few years ago, and I'm still trying to convince one of the New England aquarists to actually put a sandlands exhibit in their spaces, and which I believe would be a wonderful way of educating the public, not just about the beauty of these fish, but also of their important intermediate position between the primary productivity, the primary consumers, copepods, the planktonic basically level, and those iconic top predators that we all love to look at and fish and take pictures of. Thank you very much for your attention.
Thanks, Hans. So I just wanted to remind everybody that they can submit questions through the question box on their GoToWebinar control panel. We do have a question now that asks, have you considered exploring embryo survival under pre-industrial levels, 250 microatmospheres, to see if they improve or if they are optimized to ambient CO2 levels?
That's a very good question, and the answer is we have not. But that would be something that I believe is worthwhile exploring from a scientific standpoint. From the standpoint of what is going to happen to our ocean, it may be less important because we will not see these CO2 levels ever in our lifetime for sure. But I believe from a scientific standpoint, that is a very interesting and good question. And of course, a similar thing has been done not on fish, but on bivalves before, where the researchers were able to show that the bay scallop, when you rear them under pre-industrial levels, that they would actually respond positively. So they had benefits from these pre-industrial levels than compared to ambient levels. But that is a step in the next direction.
Okay, so the next one is, we have been regularly sampling sandlance near shore in Casco Bay area, essentially at the low tide line. What is known about the nearshore distribution of sandlance?
That's a very great question, and it is something that I'm absolutely intrigued by. We have to start out by saying that sandlands occur. There's two different species of sandlands, which are, to the untrained eye and even to the trained eye, look almost identical. And the one species that I'm focusing on here right now is the northern sandlands, Ammodytes dubius, which occurs offshore. The species I believe that the person who asked that question in Casco Bay is referring to is the American sandlands, Ammodytes americanus. And how these species that are absolutely identical in their trophic level and in their appearance, also in their spawning season, how they potentially differ in their CO2 sensitivity is, again, from a scientific standpoint, a highly interesting question. It would be a question that would effectively test this ocean variability hypothesis, because we would predict that those Americanus sandlands in Casco Bay at the low tide line would be exposed to higher variability in CO2 and therefore should be more tolerant to CO2 than those offshore Ammodytes dubius. It is a question that I'm very, very interested in to pursue in the future, and chances are that I will contact somebody from Casco Bay and ask how exactly do I get my hands on spawning ripe American sandlands.
All right, so the next one asks, can your results be extrapolated up the food chain to estimate overall impacts?
I would caution about this. I specifically and carefully worded my conclusion that our results imply concern for these higher trophic levels. I would say that we should be extra cautious in our approach for now to exploit this resource. There is some argue that the sandlands should be exploited commercially for bait or for fish meal production, which at the face of these results seems like not a good idea, because we may need every last single bit of sandlands for these iconic predators. But while I would love to entertain a modeling exercise that could show what would happen if sandlands in the next century basically suffer a 30% loss of their egg survival, I also know that that nature is very dynamic, and that we are not yet in a position to confidently announce to the public that we basically plugged in our survival numbers right now and then extrapolated that this translates into half of the whale population. I would not be comfortable doing that.
Are you aware of any studies which show nearshore or open ocean fish species benefiting from increased CO2 levels?
I am not.
No. Okay, so the next one is, did you consider using geometric morphometrics to separate the effects of size per se from the effects of lipid and yolk storage? It would be interesting to know if size corrected effects remain.
the size of these embryos, you mean, okay, that is a question that we need to perhaps discuss in a more like detail. So the short answer is I do not believe that that size was sort of like in how the review or that the questioner is imagining that we did this geometric mean. I think all we showed here is basically the sizes at hatch, the somatic area plugged into a principal component analysis. So I would need a few more details basically after the seminar to better understand how else these data should be analyzed. What I can say, however, is that yes, there is as we showed, a difference in size between these temperature treatments, but the size differences are statistically significant, but not very large. So I would defer like an answer to that question until I exactly understand how it is manned.
Okay, so this next question is actually a follow-up from a previous question, the one asking about nearshore distribution of sandlands, and you mentioned another species.
Yep.
They ask, is this species also functioning as a key forage species?
Yes, I believe very much so. The American sandlands has in fact been like investigated a few decades ago by a person from the University of Rhode Island, the late Alphonse Migelski, who looked at it already for its temperature sort of dependence. And while I would say that in the nearshore areas, American sandlands have a few other fish species that are still functioning as important forage fish, we now more and more understand that these American sandlands in general have a specific lipid composition that makes them particularly good forage fish. So if you are a tern, a roseate tern, or an Arctic tern, and you have the choice between a sandlands and a silverside, say, you would probably like to go for the sandlands. So yes, the short answer is I believe these are also very important forage fish species in the nearshore.
Great. So the next question asks, are there studies or data sets that detail observational population estimates of sandlands at any spatial scale, especially in conjunction with changing climate and ocean acidification?
I believe there is very scant knowledge about population. There's no assessment because it's not an exploited species. There's no assessment for sandlands. We do not know what actually constitutes a population. My second last slide basically, like honed in or alerted to that fact that we know so little about sandlands in terms of what constitutes a population. Are the species, is the population on Stellwagen Bank, say, a self-recruiting or self-sustaining population? It is well possible that that sandlands spawn on Stellwagen Bank, but if you have a larval duration of about 5 to 6 or 7 weeks, then chances are that you are transported somewhere completely else than the sandbanks that you actually hatched from. It is possible that sandlands on Stellwagen Bank recruit from a more sort of like upstream population of sandlands and then are swept onto Stellwagen Bank. The short answer is there's no assessment, there's very little knowledge about what constitutes a population, and therefore there is an urgent need to better understand what drives these populations. Our European friends are a little ahead of the game there. because European sandland species in the North Sea have been investigated to greater detail. This is Tobianus, Ammodytes tobianus is one of those species. I think that is the unsatisfying but realistic answer to that question.
All right, so this one looks like our final question. Says, great talk. In year 3, 2018, did you do lab-based spawning only? What were the methodological different changes between years 1, 2, and 3 that may have resulted in different hatch success? And were the CO2 effects on hatch rates at 7 degrees intermediate between the 5 and 10 degree temperatures?
The 2018 fertilization was done at sea only. We looked at this, and we basically learned the lesson from 2017 and believed that it is better to fertilize these fish right away. A methodological detail that I didn't bring up in the talk is that we had an inkling from other literature that it might be important to, after the fertilization, use diatomaceous earth, which is a very fine powder that some people actually put in their drinks. It has some cleansing, maybe properties, but in this case, it is said to prevent the clumping of the eggs. The clumping of the eggs, we thought, is a thing we should prevent in order to, give every egg the same kind of oxygen supply. We found, however, that the diatomaceous earth did not outweigh the negative effects of holding these fish for 2 days in the laboratory. And lastly, the effects of 7 degrees were intermediate between the 5 and the 10 degrees, and we have, for clarity reasons, have not presented them here.
Okay, so, well, that was our last question. So that concludes our webinar for today. The recording of this webinar will be posted to the NECAN website. Our next webinar will be on Wednesday, October 9th at 1 p.m., presented by Maura Nemisto of the University of Maine on gene regulatory response to end-century temperature and PCO2 in post-larval American lobster. Thank you again, Hans, and thank you everyone else for joining us today, and that concludes this webinar.